Many of you will know potentially Opie Jones. You might have seen his mother, Lucy, present at Meeting on the Med, I think this past year, and a couple other presentations. Opie was 1 year old when he was diagnosed with pediatric ALL. He relapsed after a bone marrow transplant, was one of the first patients of his age bracket to get, I believe it was Kymriah at that point, in the UK. And in July, he celebrated 3 years cancer-free, which is a fantastic milestone. We had Opie and his family at our team meeting in July, and he was running around tearing things off the walls like any 4-year-old you would have expected to see. It was great to see him doing that. So there's some of you in the room who won't be as familiar with Ori as others, so I want to quickly just via video introduce who we are as a company and what we're trying to do here.
We are manufacturing brighter futures. Together.
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 Together! Together!
Together.
And again?
As a company, our mission is to enable widespread patient access to life-saving cell and gene therapies. So partnering with you, the industry who's working incredibly hard to make sure patients can get access to these curative therapies. And our goal is to work with you to manufacture brighter futures for all patients. Some of you might have seen our launch at ISCT in May in Vancouver. So the IRO platform, which you see here, was launched then. We have subsequently unveiled the platform to the world. We've highlighted some data, which I'll run through quickly through our work with CTMC down in Houston. Many of you will know Jason Bach and his team. We have conducted, I think, way over 50 live demos now. I think it's close to 100. Deployed with 2 partners over the last few months who are having great results with one of them has a commercial CAR T process and others have clinical stage processes that we are working on with them. And we're getting great feedback. IRO is the easiest thing I've ever worked with, is music to our ears. We've worked very hard on usability. We worked very hard on building an intuitive platform so that we can take labor out of the process, we can de-skill the labor that's left, and really drive up throughput and drive down cost of goods. That ultimately benefits all of us and benefits patients. So the platform itself consists of the instrument, which you see here in the room, and also some consumables, which you might have seen in the area outside. And here, you'll get a chance to have a play with them later. And if you have a demo, you can see them in action. One of the novel features of the Ori platform is the bellows-based bioreactor, so that kind of accordion you see there. Very unique design. We have 36 granted patents so far around the various elements of the platform. But that bioreactor allows us to operate in a large working volume range. We can go down to 50 mil, do a genetic reprogramming or activation step at a very low volume, and go all the way up to 1 liter working volume. So that essentially means we can go from 50 million cells as patient starting material to over 12 billion cells of therapeutic product in that singular vessel. So it never has to leave the inviting confines of the IRO instrument, which is for optimal growth conditions, and it has the ability to do all of the necessary steps that are required as part of the process. The other key innovation, the Ori Connect system, allows us to sterilely connect into the bioreactor in a dirty environment. It's a closed system. So I think the industry, some of the folks in the industry have invented a term called functionally closed. It's a marketing term, it's not a regulatory term. There is no functionally closed. It's either closed or it's not. This is a closed system. We have the ability to operate in a grade C, grade D, or even controlled non-classified environment. There's no tubing, there's no bags, there's no tube welding, there's no pumps in the system. We're able to move fluids through that sterile connection system, which we'd love to show you in more detail as we go. I'm excited to have Matt here. Many of you will know Matt Hewitt is one of the thought leaders in this space. Charles River is one of the leading CMO partners for much of the industry. They're partnered with Casgevy and Vertex on one of the approved sickle cell products. And I'm excited to invite Matt up to the stage to discuss the work that we've been doing as part of our Lightspeed Early Access program.
All right, thank you all. So introduction, I obviously don't look like my picture. I half joke that I do this so people can't find me at meetings. So I'm gonna switch this picture after this talk and shave my beard. So quick overview, Charles River's been around for 77 years. We have primarily known in our preclinical CRO business, which we've been doing for all of that time, and we've recently added, probably about 3.5 years ago, substantial capabilities in CDMO. And we combine that with our testing capabilities, which we've been doing for over 20 years, as well with cell supply to create more or less an all-in-one solution. Why is that important, specifically in advanced therapies? A lot of the preclinical models do tell us a lot of things, but they are certainly not perfect. And generally, your preclinical models are where you start to establish things like your CQAs, your critical process parameters, your product profiles. And many times when you go in the clinic, you find out that those things sometimes need modification. And so then you end up having to go back to your preclinical CRO, separate group, have conversations, and try to figure out what's the best way to do that. Well, for us, it's an internal conversation. So we work directly with our preclinical partners in that business to make sure that we have the two really well connected. I would say the other thing that we do is when we start talking about CQAs, and this is certainly in cell therapy in regards to potency, but a lot of times as well in gene therapy and AAV, there are certain assays that are developed in preclinical that look very good and meet the needs for preclinical. But then when you start to transition to GMP and clinical, and you say, is this an assay which can be qualified and validated? That's a completely different topic. And we know that Peter Marks and the rest of FDA have been very clear that a substantial clinical effect alone is not going to be sufficient for approval. You have to have the commensurate analytics to go along with it. So we do focus not only on the process, but also the analytics. This is from a slide last year, Kimberly Schultz at the ICT North America meeting in Houston. This was primarily in the context of them bringing out in the open some of the decentralized manufacturing. But the point is they start highlighting the fact that they are looking at, in addition that we're getting a lot of INDs, that they want to see new manufacturing strategies. And this, to Jason's point, is one thing that we've been seeing. Now, I have a prior life where I did launch another piece of equipment in the field, and that was one of the things that they were very clear about when we were doing our meetings with them, was that they wanted to innovate in the space. And I think people in the therapeutic side, clinical side, sometimes voice a bit of frustration about this and say, why are we taking so long? Really and truly, I know we have Glybera and Provenge that were out there, a while back, and I don't want to steal their thunder, but really the clock probably started ticking at 2017 with Kymriah's approval. And so we're 7 years out from that approval. Now we're starting to see a lot of this technology come to the market, and we're starting to see some of the, problems around the manufacturing being solved. And so the fact that they're now talking about this points to the fact that they see that that's coming. So we have a problem from a business sense, which is good, that demand is outstripping supply. But from a patient access perspective, this is an absolute tragic problem for us to be dealing with. And so there are a couple examples of this. Christy Shaw did an interview with McKinsey back in early 2023, which is that within their portfolio of commercial CAR-Ts, they only address about 20% of the patients that are eligible to receive, Yescarta and Tecartus. If we look at another ASCO abstract where they surveyed 17 US clinical centers, they found that only a quarter of patients that were eligible for commercial CAR-Ts received one, and there was a median 6-month wait time for that therapy to be delivered. Another 25% went into a separate CAR-T trial, and the remaining 50 went into another clinical trial, and then unfortunately probably went into hospice and died. So we have 50% of the market that is not receiving what they're eligible for. So this is a big problem that we need to sort out. If we talk about commercial success, which is now becoming more and more of a topic in the field. So we have these commercial therapies, but are they selling? Are they being adopted? This is from the IQVIA report that was released in December of last year. And it tracks global commercial spend of cell therapies. And this line does not reflect a lack of adoption. It reflects a lack of supply. So it does fit with these other two data points. And so automation is going to be a very critical part for us to get through this, in addition to some of the other things around workforce. When we look at the things that are limiting, again, from the IQVIA report, 60% report time between the referral and the start of the CAR T treatment. This is vein to vein time. 60% problem. Next biggest one is slot availability, manufacturing. Can't get it in there fast enough in manufacturing. This is related to the processes we're employing to do these. If they're open, there's very tight restrictions on you for parallel processing. The last one, which is somewhat related, and this is always comes more back to decentralized manufacturing, but it goes back to automation, is the geographic distance that is required for a patient to travel to get a CAR T. And this is not limited to CAR T. This is going to be any of the cell therapies. And this was something that we, automation alone, decentralized alone is not going to solve. We're working through this with, training medical staff, make sure that, clinicians know what to do when they dose a patient with a CAR T therapy as an example, and then they see AEs, like how do I solve those? So when we look at automation, or where we started, traditionally in what I call Gen 1 and Gen 2, we have been open manual processing, for Gen 1 and separate analytics. Now for Gen 2, we're trying to go with, let's say generally all-in-ones for the manufacturing device, but you still have separate analytics. Done this cost study multiple times, and we did it again earlier this year in an ARM workshop in Maryland, and we found that typically when you just automate but keep analytics separate, you can get between a 35 and 40% reduction in COGS. So then what you need to start doing, and I guess my bias, sure, phase Gen 3 would be starting to integrate some of the analytics into the manufacturing process. So we start to blur the line between manufacturing and analytics. Generally, the easiest thing to do when we look up enough of these workflows is to start automating in-process sampling. Because many times in-process sampling is not used as a decision point for therapeutics, therefore it's not generally reviewable by the regulators. So if we do like in-process cell counts, in-process transduction efficiency, that would be the way to go. And we really, in actuality, don't need to do those time points. And in fact, we have conversations about clients about this all the time. But, they don't trust the machines. They want to know. They want to make sure the cells are growing. We know they have a doubling time. We know they have certain things, but they want to know. They want to make sure. So this is a way to get at that. And then Gen 4 is we need to completely get rid of the line between process and analytics. Because as I'll go over soon, manufacturing is just one problem we're dealing with at the moment. And there are downstream problems once we solve manufacturing we'll have to deal with as well. Decentralized manufacturing has been a big topic in the space, and I'll touch on it tomorrow. I have another talk at the conference. And I think there was some recent commentary about this, that it really is about reducing COGS. I do disagree with that to some degree. I think you can reduce COGS a little bit, but decentralized is more about increasing access. That's what we're having a problem with. And that's what the data points tell us. We have far more demand than supply. We have a problem with people having to drive long distances or travel long distances to get treatment. Decentralized is about bringing treatment closer to the patients. We might have some small, cost savings with that, with say, cheaper logistics, but there's probably still going to be some last mile of shipment that's happening there. So how do you do that? We do this already with decentralized testing. So we do have a decentralized testing format. We have them in Europe as well as here in the US. The first level that we take is we centralize and standardize a lot of our systems. A lot of your enterprise-wide systems, LIMS, SAP, Pilgrim, et cetera. Second level is, we start to create a standard BOM or BOE of equipment that we're going to be using. And then the last one is we start, and we have about, 12 or 1,400 of these, what we call general protocols. So these are generally qualified and validated protocols that we can drop in and run. And a lot of them are standard ones that you'd be running if you were trying to release a cell therapy. So this, if you haven't seen it, is another part of automation. This is actually a video that we show all of our new GMP manufacturing technicians. to help them understand why they have to do things GMP and why we are trying to eliminate nuance from all processing. And this is another part of providing access, because if we can standardize something, everybody in here knows how to make a peanut butter and jelly sandwich, hopefully. But I would say that if you had to explain it to somebody who didn't, this is what would happen. I don't know. I think we're going to play this a little bit. So we're not going to play the whole thing. I made Jason watch the whole thing yesterday. So
You're not making any sense. He's already ruined it on purpose. He knows how to make one.
Step 1, get 2 pieces of bread out. Got 'em. Get a butter knife and get some PB. Take 1 piece of bread, spread it around with the butter knife.
No, Dad, with the peanut butter.
I'm just doing what it says. It says take 1 piece of bread, spread it around with the butter knife.
Hold on.
Get some jelly, rub it on the other half of the bread.
No, Dad, open the jelly.
Well, it doesn't say to do that. Put the breads together on top of each other. Take a big bite. This doesn't taste like a peanut butter and jelly sandwich. Fail.
So there's 6 more minutes. They do make a sandwich, by the way, not to spoil it. But I mean, this very clearly helps you understand why tech transfer is extremely important. And if you have a lot of nuance in your process, you will have not just trouble transferring it to one CDMO or another site, you'll have trouble scaling it if you have to find the one person that can do it correctly. So this again comes back to why we need automation, why we need some level of standardization without inhibiting innovation. So again, manufacturing right now is one problem. Once we fix that, now we've got all these products we have to test. And again, this comes back to blurring the line or eliminating the line between the process and the analytics. And then once we're done with that, the next problem will be now we got a lot disposition to release all these products. How are we going to do that? And so, we are thinking proactively about this in terms of the systems we're implementing, as well as doing a lot of technology evaluations, which we'll go through one that we did, in the next subsequent slides here. But, and then of course the last one, maybe not the last one, the next one is always supply chain and logistics. And don't want to discount that, because if we don't have consumables and media, it's pretty hard to make cell therapies. So, these are all important things. I do want to remind us we're there. We're at the beginning still. Maybe the middle, depending on where your start and stop is. But the point is we are working towards addressing this manufacturing problem. So the what, the why, the how, what's next. We'll get into that. I mean, I think the thing that we want to do, and just to be clear, I mean, we're a CDMO and a CRO. And the role of a CRO CDMO, especially on the CDMO side for advanced therapies, is to know and understand the equipment and understand where that equipment fits with particular processes. So, we're going to be comparing some of the data here to another system that's in the market, which we'll go through. And I want to say it's completely normal, because any client is going to come to me and my teams and say, what's the right piece of equipment for my process? And if we don't know how they work and how they somewhat compare to each other, how am I going to tell them that? And then you're going to have clients coming in existing equipment that wants to move to new equipment for whatever reason, and it doesn't matter. And so we're going to do it anyway. So I do want to say that these comparisons do happen in the field. They're not a bad thing. It's pretty typical. And I'm not here to necessarily say, I mean, Jason can say it, I'm not. But that one is better than the other or worse than the other, and I'm not saying he would, but the point is that these comparisons are going to happen, and they do happen behind the scenes. We're just talking about it tonight in one specific use case. So we did some comparisons to another piece of equipment. Why do we do that? Just explained it, because we need to know as a CDMO about new pieces of equipment coming on the market. We work with others that are in the audience on this exact same thing. And how do we do it? I'll explain that. And then what's next, that'll be at the end, kind of before Jason comes back up and entertains us then. So what we're doing essentially is a CAR T process. And we did not want to do it with GFP. That was like the number one thing that we did not want to do, because GFP is always nice looking. We wanted to do it with a clinically relevant payload, which we chose CD19. We chose a lentiviral vector. And then we chose clinically relevant things like starting cell number, and then we set generally CQAs that you would see anywhere in the clinical process. What does that mean? We want to produce, hopefully around 2 billion CAR-positive cells. That's generally more than you need for a clinical process, but we were also assuming that we would like to see transduction efficiency around 50%, but a CQA is a lot of times around 10%. We didn't really go too deep on VCN or other stuff, although we can do that later. We really just wanted to take a look at, what are the top line results that we can get from the systems. So we ran the Ori against the Prodigy in this case, and we did 3 runs apiece. You can see that generally Ori and Prodigy were, pretty similar in transduction efficiency, but that the Ori produced more viable cells. Now, the other thing you may notice on the graphs on the right is that there is one black line that correlates to Prodigy run 2 that is more or less flat. And this is, again, stuff happens. We had a critical error on the software on that run for the Prodigy, and so we weren't able to complete it, and the cells died. This is something that happens. We're not saying it only happens with the Prodigy. It happens with any piece of technology. Happened with the pointer before you all came in that Jason and I were fighting with. So I mean, this is kind of common stuff, but this is the data we got when we did these 6 runs. So in general, we are seeing much higher cell numbers. In general, we are seeing some slightly higher transduction efficiency than the Prodigy, but we met all the key critical parameters that we had set in this case. And I would say we have more than enough those cells in this case to dose, assuming that we had a similar outcome with patient material. And that's more or less reflected here, that we're trying to create an outcome matrix where we look at, okay, what happened? Do we meet it? Do we exceed it? Do we fail it? Obviously, if you fail something, we want to look at why. I mean, and we don't say, okay, the Prodigy doesn't work. What we say is how do we adapt the process further for the Prodigy? I mean, that's the CDMO talk, and quite common. So in this one, we were able to take the Ori out of the box, so to speak, and run it on 3 runs, and it did quite well. The Prodigy did not do as well in this case, and some of it was related to the failed run that we had on the second run. So we will continue to use this. Obviously, we use both systems. We have a lot of experience with the Prodigy. We'll continue to use it, obviously, when it's appropriate, and the same for the Ori. So we're giving a preview of what's coming out tomorrow. Well, I guess one thing I didn't know, but we are talking about what's next tomorrow, which is that we're going to be continuing to work together on between Ori and Charles River to use the IRO for various projects and looking at what other data we can produce together that we can do more talks like this. In addition to that, I guess Jason and I were interviewed yesterday by Fierce, and I guess that one is also coming out tomorrow morning as well. So I guess we probably say you said something, I don't know. Yeah, somewhat intelligent, enlightening. So, we're super excited about this. Again, this is a field where we need all the options on the table, because for us, and I think everybody in the room, this is about bringing the best and the safest therapies to patients. And that does not necessarily mean we're always going to use the IRO. That doesn't mean we're going to always use the Prodigy. That doesn't always mean we're going to use the G-Rex. It means we're going to use the system that does that process the best and delivers the best results for patients. And that's why we're doing this. So I guess I'm not going to read my own statement. I'm not quite a politician yet, I don't think. Still can't watch myself on video. But you can read this and you say, again, this is what it's coming back to, the last few sentences I made, which is that the IRO did show us very, very good results in a head-to-head comparison. And what we want to do here is use technologies like the IRO to address the manufacturing bottlenecks we see in the space, because that's we have to fix that. We just have too many folks that are going without these, many times life-saving therapies. And with that, I'm going to hand it back to Jason.
Thank you, Matt. Many of you will have seen this resource that we created now a year or two ago when I asked the team, how many patients are getting treated out there? And the number wasn't readily available. So we track this every quarter. We update it based on public information. It will be specifically wrong, but directionally correct as far as the trends go. It's available on our website. So you're welcome to take a picture. You don't have to. You can find it on our website if you need it. But the good news is that we are on track to deliver 10,000 doses for 10,000 patients in 2024 for the first time. And that is fantastic news for those 10,000 patients and their families. The focus of this discussion also needs to be on those tens of thousands of patients who don't get access, unfortunately. That's really the problem that we're trying to solve. Matt quoted numbers, and I've seen those very same numbers, 20%, 25%. That is almost exclusively in the US, those numbers. There's a very limited access in Europe, very limited access in the rest of the world to these therapies. And our hope is that we can work with you, the folks in this room, to improve these numbers as we move forward. Tim Hunt, who's in the room, gives a fantastic welcome or state of the industry, usually at JP Morgan to kick off the year. Unfortunately, the status quo does often remain death or serious disability for those patients that don't get access to these incredible life-transforming therapies. And that ultimately is why we all get out of bed in the morning. So how do we end up here? I think, I have a 20-odd year history working in pharma. I was in small molecules for much of that time. When we're developing new products, we're laser focused on safety and efficacy. We assume that we can get safe and effective products, they'll get approved, they'll get market access, we can manufacture them and get them to patients. So that kind of third bucket or that third pillar of viability is assumed in small molecules, and we have now become able to do that after decades of development also in biologics. That is a core tenet. We took those assumptions into our experience starting in 2017 with cell therapies, and we made the assumption that if we could make safe and effective products, that they would be covered, we could make them, we could deliver them to patients at an affordable price, and that has turned out not to be the case. And I think the question around viability is a critical one. And I was having breakfast with one of the thought leaders in the space this morning, and she was saying, this is the critical balance, is when's the right time when we're focused on, we're all going to stay focused on safety and efficacy, of course, those are our core pillars. But we also need to focus on viability early in the process. We need to focus on manufacturability. We need to focus on CMC. We need to focus on a TPP that can deliver a cost of goods that can make this product available. Because ultimately, the investors who invest in this space require us to deliver an ROI on the investment that they're giving us. And if they don't think we can cross the viability Rubicon, they remain skeptical in cell and gene therapy's viability as a modality. And this is part of the reason why we've seen such a slowdown in cell and gene investment. Now, ARM put out some very interesting data in this quarter where we've seen about $11 billion in investment in the industry this year in the first 2 quarters. That's almost exactly equal to the amount of investment that happened all of last year. So the hope is that some of that investment is coming back. But we know that investors in this space, therapeutics investors, have focused on things like radiopharm, ADCs, bispecifics. Those areas are hot at the moment, and cell and gene therapy is looked upon a bit skeptically. And so it's up to all the people in this room to demonstrate we can create viable products that can reach patients at scale. And that ultimately was the design thesis, the foundational thought process that led to the IRO technology. So what is the IRO supposed to be doing for us? We need to automate better biology. We need to be able to deliver superior biology to get the therapeutic benefit, that safety and efficacy milestone that we're trying to hit, but we also need to do so affordably. So automating better biology, accelerating product development. We think we can get you into the clinic faster by having an automated platform. The IRO platform is GMP ready. You can do it R&D all the way through GMP in the same platform. Same instrument, same consumables, same process, same reagents. That should help save time. That should reduce comparability risk. That should reduce your overall development time, maybe by up to 3 years. If you look at those tech transfer times, if you're tech transferring once or twice during your development process, and you can go from 6 to 9 months to 6 to 9 weeks, that's a massive savings. So accelerating product development is a core tenet, and then scaling your impact. Ultimately, we need to deliver 10,000 doses and then 100,000 doses of these products to cover these larger indications, solid tumors, autoimmune disease, all these fantastic opportunities that are in front of the industry. We need to be able to deliver the benefit to patients. So what does that mean and how does it link to viability? So the IRO platform helps address the major manufacturing bottleneck today, which is the activation, transduction, expansion, and harvest steps. Those are the longest parts of the process, often 5, 7, even 10 days of a standard CAR T process, or longer sometimes in TILs or TCRs. So addressing that bottleneck opens up the throughput of the system. And then, as Matt said, we have to look at things like QA and QC next. We look at full automation, robotics, and where the other bottlenecks start to creep in, and we can then address those subsequently. Some of you will be familiar with the theory of constraints. Effectively, a system is constrained by the bottleneck, right? So if we have a process that's throughputting, you've got an hour or two up front where you're doing thawing, you might be doing selection, you might be doing loss of concentration up front, and then you're spending, that's 3 hours, 4 hours of your process, and then you're spending 7 days expanding cells. That's your bottleneck. And so ultimately, we help achieve higher cell yields, higher transduction efficiencies, which means more CAR-positive cells more quickly, shortening processes. So some of our partners have said to us, we want you to help shorten our process by a day. We've been able to deliver their CAR-positive target in 2 days fewer than they're actually doing today. So shortening processes, reducing cost of goods by using less viral vector, these are the kinds of benefits that we're starting to see, which help us lead to viability in the future. Accelerating product development, what does that mean? That means getting products into the clinic more quickly. That's a critical milestone for you as therapy developers. We want to reduce that process development time. We want to reduce that tech transfer time. Reducing variability. So we see somewhere between 20 to 40% failure rates for the commercial CAR T products to meet dose. Oftentimes they just haven't gotten enough cells in the right amount of time. We can help you reduce variability by automating the process. In today's world, we take a variable input. So in autologous therapies, we're taking a patient's starting material and we're adding process variability on top of that. With the human beings that are moving stuff around the system, shaking bags, doing tube welding, we have the ability to reduce the variability and reduce those failure rates substantially. And we also have the ability to eliminate some of that comparability risk as you go through your process from preclinical to clinical and clinical to commercial. Having to reinvent the process several times, as you all know, is a huge amount of effort, a huge amount of time, and a huge amount of resource, and introduces a lot of risk into your program. Lastly, scaling your impact. You have the ability, you saw on Matt's slide, and you'll see in a second in the video, the ability to multiplex the system. Essentially, you're driving higher throughput per square foot in your existing footprint. So we can deliver 1,000 doses a year out of 1,000 square feet of clean room space. That's roughly 30 patients in parallel. Because it's a closed system, because it's automated, we can make sure it's a zero defect system and there's no risk of cross-contamination or errors being made in the system. By reducing labor, we have the ability to reduce cost of goods dramatically. And what does that mean when we say cost of goods? So no one knows exactly what the industry's cost of goods are. We have a cost of goods model we'd be very happy to run with you. If you have a question about, we think it's going to be this or we think it's that, what does the model say? We validated this with Deloitte and McKinsey and several partners in the field. I know Matt and his team ripped it apart and we put it back together, really trying to understand what's driving our cost base. And so today's world, we're talking about somewhere between $150,000 and $250,000 a dose for an autologous CAR T product. And I've seen estimates at both ends of the scale of that range. We think the first generation IRO system, which you see in front of you, will reduce that by about 40%. Now, range of 30 to 50%, depending on your process. Most of those savings are coming from reducing your fixed costs, reducing your footprint, reducing your labor, and reducing your consumable utilization. So lower vector, less media, less activation reagents, those things that are very expensive in your process. This is the first cut. Where do we go from there? Ultimately, consumables and labor remain a very large proportion of that cost of goods. So addressing with second layer automation, we are able to address other bottlenecks and further lower the costs. We would model for this particular process a reduction to about $75K in the near term. That we think is eminently achievable. But we want these products to be first line. A product that costs $75,000 to make, we're going to have to charge several $100,000 for to make back that massive R&D investment. We need to go lower if we want these things to be first line. And ultimately, we believe that the end game is probably under 40K. We think that gets us to probably first-line therapy, very cost competitive with a transplant, and we think it's doable if we're able to really address some of those consumables, some of the viral vector costs, and others. Now, I'm not saying that's going to be easy, and I'm not saying that's going to happen tomorrow, but I think with the combination of the folks that are in this room, we should be able to achieve that goal. Part of the savings also rely on scale. Now, people ask about end-to-end processes and can you do everything? Is it a jack of all trades, master of none technology? We've been very focused on solving the bottleneck, solve the problem, and then we can figure out you have flexibility to scale. Maybe you need a clinical array. You need 4 instruments or 6 or 8 or 10 or 20. It doesn't matter how much throughput you want. You can design a system that can deliver that throughput. And then you're thinking about ballrooms. How do I separate my upstream? Maybe there's some Class B open steps in a grade A isolator, but I want to operate in C. How do I isolate that? How do I create a system that maximizes throughput for my investment? And this might be racks of IROs able to run lots of patients in parallel. First will be administered to by human operators. We de-skill the labor. We take about 60% of the existing labor out. But ultimately, full automation where you have robots ultimately take over these tasks, repetitive tasks. The system has been designed with this in mind. Now, if you come to our labs, are you going to see robot arms flying around? You're not, because we don't need, that's not a solution to a problem we have today. What we need to do today is fix the bottleneck. If we can fix the bottleneck, we increase throughput. And when we go from 1,000 doses a year to 10,000 doses a year, maybe we need to start to think about some of these things and think about how do we automate QA and QC? How do we automate some of these other critical steps? So ultimately, the science has been proven out. I don't think there's a scientific question yet still to answer. These products are safe, they're effective, and they work. And the technologies from over the last decade, the standards, the G-Rexes and the Prodigy of this world have gotten us this far. But I think ultimately, as an industry, as an enabling technology provider, we have let the pioneers down. We have let those companies that have pioneered in this space, they don't have the right tools to take us to the next level, to take us to this big ambition of delivering tens of thousands of doses to patients. They've been very public about the challenges that they have had in trying to scale manufacturing. And so ultimately, that is the purpose. That is why IRO exists. That's why we worked so hard over the last 5 years to develop the system. And we feel like IRO introduces a new standard, a new paradigm for manufacturing that should help us solve many of these challenges and take a big step forward towards achieving that big goal, which is enabling widespread patient access. Lastly, we are also going to be one of the first companies, hopefully, that applies for the AMT, the Advanced Manufacturing Technology designation. This is still in comment. I don't think it's sort of fully finalized yet, but the guidance is pretty clear. Most of you will be familiar with the platform technology designation. We think it's going to be pretty similar to that. So our hope is that this will help our partners get expedited access to the FDA review and help them move forward more quickly in the process. So this is near-term innovation the next 3 to 6 months. The next innovation around robotics, around other unit operations will come over the next 12, 18, 24 months and the years to follow. So that's where we are. That's the extent of the presentation. I wanted to invite Matt up for a Q&A. We're very happy to take questions. If anything that we have said has been thought-provoking or you want to know more information on, we'll do Q&A for about 5 minutes, and then we can go out and have a drink and have a chat. There are roving mics. We need to stay away from that. It's probably better just from a policy perspective. It's better for us to stay away from each other. Yeah. Anyone have any questions? Yes, sir. Down here in kind of middle. There's a mic coming at you quickly.
Got short legs.
One question. Viability is an issue, but what about persistence? One of the major issues with solid tumors is that while you may have active, functionally active CAR Ts, we initially provide the treatment, after several months or years, they become anergic. And a lot of that has to do with how you expand the cells, and they lose their multipotency or polyfunctionality. Have you guys checked with the IRO system whether you still retain the same functionality of the cells through a longitudinal study?
Great question. I'll kick it over to the doctor in a minute. But we built our own assays around cytokine release and kill assays using standard kind of methodologies. We're also doing a persistence assay where we redose over time. I don't have any data to show that yet. What we've done thus far is to demonstrate that the phenotypic profile of the cells is no different than the control, whatever that is. Maybe it's a G-Rex, maybe it's a bag. And so that we're not driving terminal effector cells or central memory. It's a sort of relative to the starting material and the control, it's equivalent, not statistically significantly different. Now, if we wanted to, the interesting question embedded underneath your question is potentially, could we drive them to more naive, more central memory, or more terminal effector if we wanted to? That is work we haven't done, but we have hypothesized it is possible. And so anxious to work with some partners on that are early in that phase. A lot of our partners are coming early stage clinical, and they have a process defined and they want to optimize it versus trying to test for potency and durability early in the process. But you want to add anything to that?
Yeah, I mean, we did do some deeper flow as part of this study to look at the subpopulations, and we didn't really see any shifts as expected. But the process that we used in this case, again, was sticking with the tried and true. So this was with IL-2 expansion, which is going to generally drive more CD8 terminal differentiation. If we were to go on the CD7, the IL-7/15 route, maybe and many times we use IL-23 at transduction to further retard the terminal differentiation. You can see some shifts in those. I think there's the question around solid tumors. That one's more complex. And it kind of depends on where your starting material is coming from, what your product is. In the TILs, and there's several folks in here, and Jason's worked with TILs. They are very diverse, put it that way, in that a lot of the TILs that are actually in the tumor turn out not to be reactive at the end, and so you have to be able to come through that. The polyfunctionality question is a space I spent a lot of time thinking about. In prior life, worked very closely with Sean McKay at Isoplexis. And I guess the key question there is what does polyfunctionality actually mean? Because they actually did a lot of work themselves looking at some polyfunctionality attributes are positive and some are actually detrimental. Some drive neurotoxicity as an example. So to Jason's point, I think we need to understand how we can drive towards those, but let's say we could do that and we could control it. In principle, you should be able to lower the dose if you're administering more polyfunctional cells, but still some work to do there.
Other questions?
The question is easy to answer. I'm loud enough on the microphone.
We can repeat it if need be.
What is the capacity of your system in terms of starting material, and how long does it take to get to the 12 billion?
So at the low end, we start with about 50 mils of fluid. Often that's 50 million cells. We can go higher than that, of course. 100, 150 million, somewhere in that range is most often the starting cell dose. We'd go from, let's say, 50 million cells of starting material to over 12 billion cells of therapeutic product in 1 liter. So 12 million cells per mil is the maximum yield thus far out of the system.
So your fold expansions and population doubles are really high, basically.
Yes.
And you still don't see any loss of memory or loss of cell characteristics.
And no signs of exhaustion using standard markers.
Must be some magic there.
Must be some magic, some voodoo that happens inside the IRO, yeah. I think it's a 170-fold expansion, that kind of 50 to 12 billion.
I got you.
There's a lady in the back there.
On the point of TILs being diverse, how flexible is IRO to accommodate all those diverse TIL types and being able to expand every single one of them?
We have a partner, Achilles Therapeutics, who is one of our first partners that's working in the, I think they call them clonal neoantigen space, so the TIL space. That's pretty much the limit of experience we have with TILs. We've worked with a TCR partner as well. The vast majority of our experience has been in CAR-T. We'd love to know the answer to that question by working more with TIL providers. I know Madonna's here somewhere from Obsidian Therapeutics. They have a very interesting program happening there. I think part of the design intent of this was to have it be flexible enough to flex to the needs of your process versus you having to shoehorn your process into the limitations of the technology. So we have built essentially flexibility into a framework. We're not going to let you design outside a space that's going to break the instrument, but within that, you have the ability to decide, do I want to rock at a certain speed, or do I want to compress at a certain amplitude? Is there an angle I might want to operate at? At what temperature do I want to set it? Have the ability to control all those parameters without the need for us to come and intervene in any way. You have complete design flexibility inside of that framework. What we heard back from the industry is some of the systems that exist today are very inflexible from a software perspective, and it takes weeks and it's very expensive to get some tailor-made changes happening. So in this case, you're able to tailor the software however you might want it, because you're the expert in TILs and your process. So we want to give you that, empower you to be able to make those changes and tweaks, and we'll help however we can. Maybe one or two more questions. I don't want to be standing between you and food and drinks. That's not usually a good place to be. Anyone have one additional question before we break? All right, thank you so much for coming, and it's really appreciate your attention.